In this one-step metabolomics procedure, biological samples are prepared for analysis of carbohydrates, organic and amino acids by gas chromatography mass spectrometry in a single procedure. The samples are first treated with urease enzyme to remove urea. The second step of the procedure is to dehydrate the samples in the presence of a detergent comprised of two volatile components, triethylammonium and trifluoroacetate. This is followed by derivatization of the components by trimethylsilylation and finally analysis by gas chromatography mass spectrometry. Automated quantification of carbohydrates, organic and amino acids, isoglycines and amino sugars allows confirmation and follow-up of most genetic disorders found on neonatal screening and an additional 38% of disorders not currently screened for by tandem mass spectrometry.
Hello, I'm Jim Shoemaker at the Metabolic Screening Lab at St. Louis University. I'm here to demonstrate one-step metabolomics, a method for analyzing biological fluids for the intermediary metabolites without separating acids, bases, and neutral compounds. This makes it possible to follow up tandem mass spec neonatal screening with a single procedure that allows you to analyze for organic acids, amino acids, carbohydrates, carbohydrate derivatives, and acylglycines, all in one step. The upshot of it is that in the graph behind me, you can see that the diagnoses in red can be made using one-step metabolomics, but would not have been picked up on current neonatal screening by tandem mass spectrometry. So let's start the demonstration.